Welcome everyone to Solutions with Insight. This is part 2 of our COVID-themed webinars. Last month we showcased 4 projects inside the Institute, and we're very delighted to bring you 4 entirely different projects today. An important theme across all of these projects is agility, the being able to apply existing knowledge, technologies, and approaches to tackle new questions. And this is really due in part to the collaborative and cross-disciplinary nature of the Koch Institute. And so just for those of you who don't know, the Koch Institute for Cancer Research is MIT's Cancer Research Center and brings together biologists, chemists, engineers of all stripes, really, to develop new insights into cancer and new technologies and tools to detect, monitor, treat, and prevent disease. It's really a model for convergence in the sciences rather than siloing by discipline. We're siloed by the problem and united by the problem of cancer. And so that wide-ranging expertise really makes for a dynamic, collaborative work environment and a shared sense of responsibility, which has really been key in moving quickly to mount a response to the challenges of COVID-19. So just to introduce myself as well, my name is Salil Garg. I'm Charles W. and Jennifer C. Johnson Clinical Investigator, which is a program to bring a clinical perspective into the Institute, again, in that spirit of convergence to really bring people together across disciplines to solve problems. So each presenter today will tell you a little bit about their project. While they're speaking, you can use the Q&A interface at the bottom of your Zoom screen to type in questions for the presenters. We will answer a handful of questions at the end of each presentation, of course, but unfortunately, we don't have time to answer them all. Thank you in advance for your understanding and for your thoughtful questions. So you can also see from the blinking red light that today's program is being recorded and will be posted on the KI website for viewing afterwards if there's something you want to catch up on. So without further ado, let's begin. It's my pleasure to introduce our first speaker today. Our first speaker is Dr. Mike Yaffe. He's the David H. Koch Professor of Science, Professor of Biology and Biomedical Engineering at MIT, also an interventionist and trauma surgeon at Beth Israel Deaconess Medical Center, co-director of the Acute Care and ICU section at Boston Hope, and also the director for the Center for Precision Cancer Medicine at the Koch Institute as well. So he will be speaking with us today about his lab's project in improving emergency care for patients experiencing acute respiratory distress. So Dr. Yaffe, take it away.
Thank you very much. Thank you very much, Salil. I'm going to share my screen. I hope this works. And hopefully everyone can see. It'll take just a moment to come up. Hopefully you should be able to see my title slide. And what I'm going to talk about today is an old drug for a new disease. Now, since I'm the first speaker, let me just remind you that we're in the midst of an unprecedented viral pandemic due to this beta coronavirus, which has been named COVID-19. And this slide, I hope you can see my cursor, is just to indicate that there are now over 10 million cases, of which about 3 million are in the United States. And we've already had well in excess of 100,000 deaths. What complicates this disease is the fact that there is currently no effective vaccine. And furthermore, we don't have any effective therapies, although there may be some benefit from a common steroid, dexamethasone, but the paper on that still, as far as I'm aware, has not been published. What's particularly trying in the care of these patients is the fact that there seems to be an overwhelming need for admission of many of these patients to the intensive care unit, many of whom require the presence of mechanical ventilators of the type that you can see shown here. And that's because one of the hallmarks of COVID-19 is the presence of a pneumonia, which, as I'll allude to in a moment, seems to be rather different than the types of pneumonias that we're used to dealing with in the intensive care unit. And this is a chest x-ray of someone with COVID-19, and you can see that the normal airspace, which would be this black color down here, is largely replaced by what looks like whitish coloration, white mass on this x-ray. Now, this is a complicated slide, but I just want to focus your attention on this part of it here. What appears to be happening after infection is the lungs, the alveoli in the lungs begin to fill up with proteinaceous fluid, And in addition, we know that there's inflammation not only of the lining cells of these alveoli, but also the cells of the blood vessels that are responsible for oxygenating the blood, but based on the air that's supposed to be in these fluid-filled alveoli. Now, there's something funny about this disease that we noticed in the ICU, and that's because it doesn't seem to behave like the typical type of respiratory failure that we're used to taking care of. And that became evident when we started to look at the values that we saw, the pressures that we saw in the patients that were receiving mechanical ventilation for this type of COVID-19 pneumonia. Now, there was something funny here, and this I promise you is the only equation I will show you. But the pressure that we measure in the airways, you can think of much like an RC circuit that some of you probably learned about when you were here at MIT. And that is the pressure that we see in the airways depends on the airflow and the resistance to airflow, but it also depends on the elasticity of the alveoli. So that the combination of pressures that we measure depends on both the airflow and the pressure that exists in the lungs, because the lungs typically become stiff in ARDS. What's puzzling here is that on the right side, I'm showing you classic severe respiratory distress syndrome that we're all used to taking care of. And the hallmark of this is the fact that the lungs are stiff. They have a high elastance and a low compliance. They're heavy. And however, we can recruit more and more lung if we just play with the ventilator. What's unique about COVID-19, shown on the left, is in fact, the lungs are not stiff. The lungs have a low elastance. They're very highly compliant. And we have an inability to be able to recruit lung volume by many of the measures that we do that typically work in traditional ARDS. Now, this is a worst case scenario slide, but this is a paper that was published out of New York. And I want to draw your attention to the last line here, which said that the mortality for people who required mechanical ventilation was almost 90%. Now, it's not that high in most places. It's about 30%. But that's still quite a high mortality rate for people that need ventilators. So what's happening here? What's different about this kind of respiratory failure? And the first clue came from the fact that patients also developed cardiac arrhythmias, and they began to display evidence of something abnormal in the blood vessels themselves, particularly those that feed the heart as well as the lung. And in fact, in this reprint from a news article from the LA Times, you can see it says a mysterious illness, a mysterious type of blood clotting seems to be happening in these patients. In fact, what we noticed was that large vessel strokes turned out to be remarkably common, particularly in young people. Furthermore, there was a very high incidence of blood clots in the deep veins of patients who had COVID-19, suggesting an underlying thrombotic coagulopathy. In fact, in this paper from the Netherlands, you can see the incidence of blood clots was nearly 30% in patients who were critically ill with COVID-19. And in fact, that blood clotting represents the crosstalk between inflammation in that early viral phase and the normal host inflammatory response. In fact, it's what we're now calling immunothrombosis. Now, we've known about immunothrombosis previously, but there has never been, in my opinion, a disease where immunothrombosis was as prominent as it is in COVID-19 induced pneumonia. And that's because it's the cross-section of where the immune response, the innate immune response, talks to the blood clotting pathway. This idea that led us to propose that, in fact, maybe part of the reason for the respiratory failure with normally compliant lungs was because the blood vessels within the lung might be blocked by clot. And that led us to propose the idea that maybe we could use an old drug, tissue plasminogen activator, as a novel treatment when other therapies weren't working. Well, is it really true that there are blood clots in the lung? This is a paper out of China from autopsy results, and you can see they describe it here about blood clots that are present within the lungs of this autopsy specimen. The same thing was seen in Italy. They called it obstructive thromboinflammatory syndrome, or microclots. The same thing was seen in Brazil. You can see the blood clots here in the blood vessels, and they called this fibrinous microthrombi. And the same thing was true here in LA, where they noticed the presence of blood clots in the medium and microcirculation of the blood vessels that supply the lungs. Now, ordinarily, blood clots, when they form, are characterized because if you measure the stress that a blood clot will support, you find that the clot over time forms, it has a certain amplitude, and then it decays as the clot lyses. This shows you the results from patients with COVID-19. This is a paper out of Italy, and you can see there's this tremendous amplitude. These clots that these patients are forming are remarkably stiff. Furthermore, I'd like you to notice that the clot does not seem to break down very much. So it suggests that although there is some clot lysis, there's a problem because the clot is too strong and not breaking down. Now, ordinarily, you and I form blood clots all the time. And we lyse those normally because there's an enzyme called plasmin whose job it is to break down the fibrin clot. And the activity of plasmin is controlled by two factors: TPA, or tissue plasminogen activator, that promotes its formation, and Pi1 that inhibits its formation. Now, when plasmin is active, it breaks down the clot, and that releases clot, a particular component of the clot, which we're going to call D-dimers. And one of the things that was most remarkable in patients that were infected with COVID-19 was they had very, very high D-dimer levels, 100 times higher than we see in ICU patients. And in fact, the higher the D-dimer level was, the larger the mortality was. It was a biomarker for death. I'm going to skip these slides for the sake of time and simply tell you that we think that that indicates that although there is clot lysis happening, it's not efficient enough to clear all the blood vessels. Well, we know from old studies for strokes and heart attacks that you can enhance the ability to lyse a blood clot if you give a drug called tissue plasminogen activator. So, and I've listed for you here several of the drugs, most of which are made by Genentech, and I'm going to focus here on alteplase in particular. This led us to propose that maybe we could administer TPA to patients with COVID-19 ARDS, and that would help to clear the blood clots in the lungs and improve pulmonary gas exchange. So a bunch of us got together and started a clinical trial. It's done here at Beth Israel Deaconess Medical Center and at University of Colorado at Denver. These are the people that are running the trial. And we did an observational trial first. We started with 6 patients, we increased it to 12, and we gave patients with respiratory failure a small dose of TPA and looked at what happened. And everywhere you see an arrow, that's where the patients got TPA. And on the y-axis, I'm showing you a measure of blood oxygenation. I'd like you to notice that every time they got TPA, their blood oxygenation got better in this observational trial. Sometimes it got worse again, but it consistently got better after they got TPA. And this now led to a phase 2A trial that we're actively trying to recruit patients into now. This is a rapidly adaptive trial. We don't need many patients. We think we can do everything with 50 patients, and we can figure out what's going on pretty quickly from only 10 patients. It involves giving doses of TPA at 2 different doses, and then following a bunch of lab tests and seeing how the patients do 24 hours later. Now, the problem, of course, is that by the time we got this trial launched in Boston and in Denver, that first wave of the epidemic has passed. And in fact, we don't have enough patients right now to be able to gather this information quickly enough. And so we're actively trying to recruit from hospitals and centers in Florida, Texas, Arizona, and Alabama. And I would urge any of the viewers of this talk, if you know intensivists in those cities, please ask them to reach out to us so that we can perhaps enroll them in this phase 2A clinical trial. I will stop there and I will take any questions. Thank you.
Okay, thank you very much, Dr. Yaffe. That was great. I actually would love to start with a question of my own, which is just, are you guys at all worried about conversion to bleeding or hemorrhagic events as a complication of the TPA?
It's an excellent question. And in fact, this has been a barrier in some ways to our ability to get the trial done. We know that in patients with strokes or heart attacks who get TPA, the bleeding risk is about 1%. Now, I'm going to share my screen one more time to make one point, and that is that bleeding risk of 1% doesn't bother us as surgeons. And it shouldn't bother, I would argue, pulmonologists either. Because the alternative therapies are things like ECMO, extracorporeal membrane oxygenation, and there the bleeding risk is somewhere between 10 and 30%. But in fact, what we found is that many pulmonologists are very hesitant to allow us to use TPA because of this 1% bleeding risk. And I think that reflects the difference between surgeons who are used to dealing with bleeding all the time and pulmonologists, most of whom are not so used to dealing with bleeding. So I think that's actually a cultural shift. But I would say if with a death rate of 30%, I think a 1% bleeding risk is certainly something that pales in comparison.
Okay, very, very nice. Perhaps sort of a related follow-up is, are there current recommendations for anticoagulation in phases of COVID-19, and how does this interface with that?
That's an excellent question. And in fact, there's a lot of controversy at the moment about whether to anticoagulate these patients or not. At some of the hospitals, including ours, we're very aggressive about anticoagulating these patients because we see all of this evidence of strokes and deep vein clots. Other places, however, don't feel that way. And there are several ongoing clinical trials right now investigating whether patients do better if they're anticoagulated or not. In our experience, those high levels of D-dimer suggest that there's ongoing clotting at a tremendous rate. And therefore, we very aggressively believe that almost all of these patients should be anticoagulated in one form or another.
Gotcha. And so this would sort of build on that treatment and be consistent with it.
Absolutely.
Could you maybe comment more on what do you think really makes COVID-19 so different from a clotting perspective, in addition to this kind of plasmin type mechanism? And, is there more to this beyond resolution of the clot?
So a lot of this is driven, in fact, by the cytokine storm that we see that characterizes the early inflammation in this disease. One of the main cytokines that seems to be responsible for this is the cytokine IL-6. And among the things, although we think about IL-6 as something that regulates the immune system, in fact, IL-6 is responsible for upregulating many of the clotting proteins, and as well as many of the proteins that block clot lysis. So I think what we're seeing is really the consequence of massive amounts of inflammation, because this virus in some way has completely hijacked the normal immune system. And so that's why I say that this is an immunothrombosis. Not that we haven't seen immunothrombosis before, but the fact that this virus is so good at driving a cytokine response has, as a consequence of that, basically inactivated our ability to limit clot formation.
Wonderful. And maybe one last question is if you could comment on how good do you think the D-dimer biomarker is in general, and do we need better biomarkers for predicting risk in COVID-19 patients?
We definitely need better biomarkers. D-dimer, I think, is a useful, it's obviously been proven to be a reasonable prognostic marker. But I think where it's really helpful is it tells us something about the overall clot burden that a particular patient has. But certainly, we definitely need better biomarkers, and I suspect that my colleagues whose lectures will follow mine will tell you a little bit more about some of the biomarkers that they're trying to develop.
Wonderful. Thank you so much, Dr. Yaffe. That was really enlightening. I learned a ton from it myself. So with that, we will go ahead and move forward. So our next speaker is Dr. Angela Kohler. Dr. Kohler is the Samuel A. Goldblythe Career Development Professor in Applied Biology. Her laboratory is well known for building chemical tools and technologies for innovation in cancer therapy, some of which I must say we use in our own lab. And she will discuss how she's using these tools and has adapted them to develop novel therapeutic candidates for COVID-19. So, Dr. Kohler, whenever you're ready.
All right, hopefully you can see my screen. It's one of the best parts of my job is to have colleagues like Mike Yaffe on my floor. And today I have the opportunity to tell you about how our labs have come together in the era of COVID-19 to try and think about development of novel antiviral therapeutics. And so to start, I'm actually going to take you back to March. Oops, not advancing. To March 11th, which was a very, very dark day. It was probably one of the darkest days for me here since I've been at MIT. This was a day after we had asked our undergraduates to relatively rapidly pack up and leave the campus. A number of the PIs were starting to think about how do we shut down our laboratories and basically in a safe way, but also to sort of keep the mindsets of our students in a good place and have them think about research and important problems moving forward. And so we met here at this stairwell that I'm showing you next to the MIT Center for Precision Cancer Medicine bulletin board. I was about to leave the building, ran into Mike, and you know, we started to talk about our classes. I mentioned that I had just given a case study on repurposing in remdesivir. He was teaching biochemistry to undergraduates and mentioned that he was teaching about the different targets that were emerging for COVID-19, including different proteases and the mechanistic enzymology. We started to exchange some tips associated with the shutdown of our labs. And we just kind of looked at each other and said, we should do something. Like, let's combine biochemical expertise and chemical expertise and try to do something here. So what did we know on March 11th? We knew that there were over 1,000 SARS-CoV-2 genomes that had been published. There were several crystal structures of the different viral proteins that were emerging that were either solved or crystal structures were en route. And that included the spike glycoprotein that many people are familiar with, different members of the RNA replication machinery, and also viral proteases. And at that point, we knew a little bit about different therapeutics that were starting to emerge in clinical study, including remdesivir. So we had a group of people in our labs that decided to form a team. We had Suzanne Swartwout and Daniel Lim from Mike's lab, who are amazing biochemists, and Becky Leifer in my lab, who has deep expertise in small molecule screening, and Bryce Curtin, who's a synthetic chemist with expertise in docking and virtual ligand screening. And so as a group, we decided to focus our initial efforts on the main protease, which processes different non-structural proteins of SARS coronaviruses to yield functional viral proteins. This is a target that's called either MPRO or sometimes 3CL-Pro. I'll refer to it as 3CL-Pro throughout the talk. And we thought it was an intriguing target at that time, in part because it's essential to virus replication. It has unique cleavage specificity relative to human proteases, which means that you have the opportunity for a reasonable therapeutic index moving forward. There are multiple crystal structures, and right here I'm actually showing you a structure that was published in Science actually in April. So that crystal structure enables us not only think about how to develop a high throughput screen in the first place, but also using the principles of medicinal chemistry, how would we optimize our hits moving forward? There was a precedent for inhibition of 3CL proteases using small molecules, and in fact also for the SARS-CoV-2. A number of people have used machine learning approaches and wet bench approaches to screen now for this target. And then also molecules that we find against this critical protease are not only potentially useful in this current outbreak, but could be useful downstream if there are future coronavirus outbreaks as well. So we decided as a team to take a combined in silico screening and wet bench binding screening approach. Bryce Curtin in our group stayed at home, and he employed an in silico docking strategy using the GLIDE programs through Schrödinger. He started with 50,000 lead-like compounds that are pre-competitive molecules housed here at the Koch Institute. And we decided to focus on this set of 50,000 as opposed to billions of molecules, because if we found hits, we knew that we would be able to rapidly find those stock solutions and compounds here and move forward with them. And then Dan Lim in Mike's lab expressed the protease and worked with Becky Leifer in my lab, and they came into lab with very limited time and executed a subset of binding screens against the same set of 50,000 lead-like compounds, as well as a set of 6,800 molecules provided by the Broad Institute. It's a drug repurposing library. This was minimal work in lab, and this also enabled the team to actually go deep on the data analysis from home. And then we take these compounds, prioritize them in a secondary binding assay, and then punch them into downstream protease activity assays developed by Dan in the Yaffe lab. And so the goal here is if we get actual protease inhibitors, we would push them forward into functional studies such as replication assays biophysical and structural studies like crystallography, where do these compounds actually bind, and then use the principles of medicinal chemistry to optimize them further. And so the technology that we use for the binding assay in my lab, if I can get this movie to work, it worked earlier, is the small molecule microarray assay. It looks like it may not play here. It's a low-tech distributable binding assay where we basically take compounds in stock solutions and then, oh, here we go, robotically array them onto a series of chemically derivatized glass microscope slides. And so the robot comes in, it picks up picoliter volumes of these drug stock solutions, drops them onto these slides, they react with the surface. This yields 100 micron diameter features on these chips. We can put tens of thousands of compounds on these chips. And then what we did was we basically came in with a purified tagged version of the protease from Dan and used a fluorescent-based readout to identify any putative protein small molecule interactions. Okay, and so this is the data from that screen. What you're looking at here are just DMSO empty solvent in gray, a bunch of non-hits here in blue, and then hits, assay positives, high signal outliers across 2 replicates shown in green. And so we had a number, over 200 different assay positives and a very favorable hit rate, and we're really happy with the quality of the screen. And now here you're looking at just the hits that emerged from the screen. Becky went home and used a script that we developed to evaluate the specificity of the binding signatures for the protease versus a bunch of unrelated targets. And what you'll see is that all of the spots in yellow correspond to compounds that only bound to the protease and not any of these other unrelated targets. So we filtered out a number of these compounds here in blue. 3 of the SMM assay positives also overlapped with the virtual ligand screening effort. And I'll say that 5 of the compounds that emerged in this selective hit set also came from the repurposing library at the Broad. So we took 167 of these assay positives from the microarrays, and we took 51 of the virtual ligand screening positives and moved them forward into another assay called the thermal shift assay. And what this basically measures is an alteration in the melting temperature of the protein in response to compound. So a compound could destabilize the protein or it could stabilize us. And this is usually an indication of binding. So we, as a first pass, decided to run a single dose, 20 micromolar, 218 compounds from the last screens. And I'm showing you here, 14 of the compounds shifted the temperature by more than 1 degree, which is usually a cutoff that we use for a number of screens. Shown here is a protease inhibitor that we used as a positive control, GC376, which had inhibited other 3CL proteases, and in May was also reported to inhibit this specific protease with a potency of about 30 nanomolar. So this is we may actually dig a little deeper in past this 1 degree cutoff and actually evaluate some of these compounds in an additional thermal shift assay. But the idea is that we would take these 14 compounds, maybe an additional subset, and push them into the protease assay that Dan has now developed. And now that we're back in lab, at least in a limited capacity, it's easier for us to advance these compounds. And we don't want to just stop with this protease. While we're moving that forward, we've been thinking about additional targets. And so Dan is particularly interested in trying to find compounds that would disrupt a really critical interaction for viral entry between the receptor binding domain and then also the human angiotensin converting enzyme. And this is, again, guided by good structural information, a precedent for inhibition with both peptides and small molecules at this point. And it will enable both of our labs to develop a novel protein-protein interaction assay that we've had an interest in for some time. I'm particularly interested in screens focused on a transcription factor that's encoded by ORF3b. In May, Japanese colleagues published disclosed that this particular transcription factor has potent anti-interferon activity. And we think that this target fits in well with our lab's historic interest in trying to go after traditionally undruggable oncogenic transcription factors, as well as additional programs we have around pro-inflammatory cytokines. And then finally, the teams are spending a lot of time trying to weigh the pros and cons of a larger subset of the non-structural proteins within the virus, shown here. I won't go through this in any kind of detail, but we're really trying to think about other collaborative opportunities. Our labs have a historic interest in RNA binding proteins, so we may go after some of those proteins as well. So with that, I'd just like to close by saying we haven't been really funded for this project thus far. So we've benefited from resources internally at the Koch Institute through the CPCM, the Kathy and Kurt Marble Research Fund, the discretionary funds that we've used to really launch these screens, as well as the support from the Broad Institute in terms of using the repurposing library. So with that, I'd be happy to take any questions.
Awesome. Thank you so much, Dr. Kaur. It was really fascinating. I was wondering if you could give us a sense, just given the pace of things that industry and regulatory communities are moving, how quickly do you think we might be able to get some of these compounds translated into the clinic?
So it will take time. We're at a very, very early stage here. We're just screening at this point. We would need to basically not only optimize any hits to have better potency, we'd have to think downstream about pharmacology. In our experience, at minimum, this would take 18 months, to be totally frank. And so that's why part of our perspective is that this is not necessarily likely to make an immediate impact on the current outbreak, unless we have luck with some of these repurposed therapeutic compounds. And so we have two compounds in particular that we're interested in advancing, but I think the really interesting molecules, I didn't mention this, but the really interesting molecules that emerge from the screens turn out to have chemical scaffolds that resemble HIV protease inhibitors. So it sort of suggests that we're on to something.
Awesome. Folks, just a reminder, you can type questions into the Q&A function at the bottom of your screen. Please don't be shy. I promise you, none of us are. Maybe as a follow-on question to that, could I just ask, for some of the compounds that you already have a sense of from this kind of in vitro work, what do you think the sort of bioavailability would be, or how well they'll sort of translate into accessing the lung and other potential sites where they need to get to for COVID?
Great question. I cannot address the accessing the lung at this point, but again, there are two compounds that have emerged from the repurposing library. So these are examples of compounds that have already been investigated clinically. Certainly about half of the compounds that we're looking at, half of the structures that we're looking at from the novel structures, the novel but pre-competitive structures, like I said, resemble existing chemotypes. They're likely emerged from biased libraries inspired by HIV proteases. And so I'll use the language that a chemist would use. They're tasty scaffolds, meaning that we think we could do some damage around them in terms of optimizing some of the physical chemical properties, and they're very likely to have good pharmacologic properties as well.
Awesome. One of the questions from the audience is, maybe you could highlight for us some of the key differences in the approach here to identify these compounds, and compared to sort of other work that was also screening for compounds looking at the COVID proteases.
That's a great question. So this is a target. The target that I mentioned that for the screen here is now being pursued broadly by many, many labs. There are groups in California and industry that are trying to take advantage of cysteine residues within this enzyme and develop covalent inhibition strategy approaches. Some of this is pure structure-driven, good old-fashioned medicinal chemistry. There are other groups using high-throughput screening strategies of a different flavor, and also groups running much broader virtual ligand screens on the order of billions of molecules. In our lab, again, we wanted to do something. We wanted to focus on a small subset of compounds that we had access to here and that we could advance very, very rapidly, meaning compounds that we already had in-house. We've noticed that during the time of COVID, it's been very challenging to order a number of reagents, and we did not want to have to rely on the wider pharmacopoeia and then just sort of be stuck while we waited 14 weeks for compounds. And that's happened for us in a non-COVID era. So I think it turned out that it was the right calculation for us to really focus on a smaller subset of compounds.
Wonderful. And maybe just a last question that's somewhat related is if you could just comment on the specificity then of, you talked about screening across, or there's a number of proteases and efforts across sort of many similar molecules. Are there ways we can really hone in on COVID-specific inhibitors, or maybe that's not even the right goal? Would love to hear your thoughts.
That's a great question too. So at this early stage, we've evaluated specificity against a set of 30 different proteins, none of which are other proteases. And so I think what we would want to do is in our critical path at some later stage, evaluate any leads against other proteases as well. So it's a great question.
Wonderful. Thank you so much. It was fascinating. And it's great to see how much you're able to build just with everything that's in-house. It's really, really fabulous. So I think we can move on to Dr. Sangeeta Bhatia. And so Dr. Bhatia is the John J. and Dorothy Wilson Professor of Health Sciences and Technology and of Electrical Engineering and Computer Science. She is also the director of the Marble Center for Cancer Nanomedicine and a proposal selection co-chair for the Task Force on Massachusetts Consortium on Pathogen Readiness Proposals. Has been very involved with the consortiums forming in the city. So we're excited to hear her talk. She'll be speaking about nanomedicine for monitoring, prevention, and therapy in COVID-19. Dr. Bhatia?
Great, thanks, Salil. Can you hear me okay? I'm going to assume so.
Absolutely, we can hear you.
Great. Hi, everyone. Well, this has already been a treat this afternoon to hear from my colleagues, Mike and Angela. It's so wonderful to be part of a community that is so nimble. I mean, we think we're all learning and trying to serve as quickly as we can. So I'm going to share with you kind of our perspective, which, as you've heard, kind of sits upon an ongoing approach of the KI. And in our case, the ongoing approach was one where we were taking a convergent approach to cancer to sort of meld tools of nanotechnology with cancer research and a collaborative approach in the form of the center and really repurpose some of these tools to COVID research. So the approach of the center that I run is the Marble Center for Nanomedicine, is basically to exploit materials and approaches that have emerged from the semiconductor revolution, which you see here listed on the left, that has given us sort of nanoscale materials to manipulate biology. And what you see here on the right-hand side of this molecular ruler is sort of the length scale of different biological processes that we've historically been interested in manipulating, again, for cancer research. And you'll see, actually conveniently that a virus is right here around 100 nanometers in size. And so just as for cancer research, actually, many of these tools turn out to be incredibly useful, potentially useful for thinking about virology. So I'll speak on behalf of our work, and you're going to hear Bob Langer next, but the rest of my colleagues here are also thinking how to repurpose their nanotechnology tools in infection. So in our lab, we've taken a three-prong approach to this. I love the way Angela described the sort of dark day of March 11th. That was a dark day for us too. I would say these 13 weeks of having the lab closed have been like the longest I've ever had my lab closed. And so we've been trying to move forward in that time, and we're thrilled to be back in lab and getting the first data on these projects actually in hand in the last couple of weeks. But I'll show you the three projects that we stood up. One is on the sort of diagnostic prognostic side, following on some of Mike's comments on what we might be able to measure in patients to see whether those clotting processes are going on and how much inflammation is going on, and adding to sort of the current landscape. The second project is around what we call prophylaxis, which is trying to inhibit the establishment of infection. And then the third project is around therapeutics, trying to interfere with some of that inflammation that you heard about. So this is the first project that I'd like to share with you. And it's really based on a project that we had stood up for cancer detection. And in this project, what we do is we use nanoscale materials. So you're looking at a drawing of a polymer here that's about 20 nanometers in diameter. And this material is decorated with peptides that are susceptible to cleavage by enzymes called proteases, like the ones you just heard Angela talk about. And we had been making these materials and administering them in models of cancer to detect proteases that are involved with cancer progression. And as you've heard from Angela, that obviously there are now proteases that are involved with COVID, and one can sort of repurpose this technology to detect that protease activity and get a sense of viral activity, not just sort of the RNA burden in viral infection. And so the way these sensors work is that when they encounter the protease that they're designed against, this peptide gets cleaved and a little reporter fragment comes off. And as engineers, we can design these reporters so that they can be measured in different ways. Here you're looking at them measured by mass, but we can also measure them, we can measure them a whole bunch of different ways, what I'll show you on the next slide. Okay, so in this version of the technology adaptation, you'll see that we envision that we would administer these sensors to a patient that potentially had COVID-19, and they would encounter the protease to which they'd been designed and liberate a fragment. And the fragment could, on the one hand, come out in the urine because these fragments are so small that they can be cleared by the kidney. The kidney is a 5 nanometer filter. And then with that urine, you can actually run a point of care test like a paper test or a pregnancy test. On the other hand, you could design these so that the liberated fragment is actually a gas, a volatile gas, and you can breathe it out like a breathalyzer. And so these are projects that we've stood up, as I said, in other disease areas. Leslie Chan in the lab is working on the breathalyzer, and her paper is about to come out in a non-virus application. And Melody Anatar has been working on viral pneumonia actually before COVID came, and now is pointing this tool at coronaviruses. And one thing that's been really interesting and exciting is not just the ability to monitor viral proteases as a sign of viral replication. As Angela mentioned, there are these two very interesting viral proteases that the virus uses to assemble itself. But in addition, there's a whole bunch of host proteases. So the response to the viral infection that one can measure. So here some of them are listed, and importantly, one of them that's involved with blood coagulation, thrombin, is a gorgeous protease for us to measure an activity for, and we have an existing sensor for that. And so I think, Michael was alluding to D-dimer earlier as a measure of a clot burden, because it basically tells you how much clot is falling apart. And thrombin actually is the key enzyme that drives the deposition of fibrin in clotting. And so we think this might be a really interesting signal to add on top of the proteases that we were otherwise measured that would be more immune related in this sort of immunocoagulation axis that he described. So that's the first project, and this is moving forward. We have the first data emerging from these studies just this week. Well, I keep you posted on taking that forward. The second project is one that is a collaboration with Brad Pentalute, and this has to do with the idea of using a nanomaterial scaffold to present peptides, again, a different peptide here, in multiple copies. And to try and make inhibitors. And we as a community are well aware that when you present peptides that have relatively low affinity in multiple copies on a core, you can increase the affinity of that interaction. We call that multivalency. And so the idea here is to basically make what we would think of as like a nano sponge to inhibit the interaction of the virus with the receptor ACE2 that Angela showed you. And to try and inhibit infection. And we sort of envision that this would be like a nose spray or a mouthwash, potentially moving forward. So this is just a blown up picture of that interaction where this ACE2 receptor interacts with what's called the spike protein on SARS-CoV-2. And our collaborator, Brad Pentalute, who's here at MIT in chemistry, just as the pandemic was coming into Boston, had published the description of this particular peptide based off of the structure that Angela showed, the SBP1. And this is a nice affinity for this interaction, but we'd like to increase it. So Tahura Samad and my group is working with Brad on making a multivalent inhibitor using that peptide. And we've been able to stand up a collaboration with Lee Gerke, who's in IMES, who's developing an entry assay for the virus to see if these work. And again, so these early days, we're just getting started, but we're excited about the potential for this work. And then the third idea leverages an ongoing collaboration that I had with a colleague in San Diego, Mike Saylor at UC San Diego. And this is a therapeutic approach. And the idea here is to use RNA interference to silence some of the transcription factors that drive that inflammation in the pathologic COVID-19 that you heard about. And so here what we're doing is using nanomaterials that Mike's lab had invented and we together had used actually in bacterial pneumonia previously now to try and adapt them for viral pneumonia. So the way this works is these are nanomaterials that you inhale, so they're aerosolized. They have a porous silicon, a degradable core that you load with interfering RNA against a transcription factor. We're using a transcription factor called IRF5, which is kind of upstream of IL-6 and all those other kind of inflammatory cytokines that you heard about earlier. But I think it would be very interesting based on what Angela was just saying also to look at the ones that she's identified. So you load up this nanomaterial with this siRNA and you coat it with a coating that's what we call a fusogenic coating and a targeting ligand. And so when this is given into the lung, this can find the cells it's intended for, which are macrophages in these cases that drive that inflammation, and fuse with the surface of that cell, and then dump that interfering RNA into the cytoplasm where they can then silence that transcription factor that drives inflammation. And Mike's group had shown that when you do this in mouse models of bacterial pneumonia, actually you're not touching the infection at all, but you can improve the survival of animals just by silencing the host response to inflammation. And so we're very curious now about using this potential approach in COVID. So that's kind of a whirlwind through how we've been thinking about it. And I hope you kind of appreciate the really straight kind of intellectual connection that you can make between nanomedicine for cancer and nanomedicine for infection. Really the nanoscale here is allowing us to sense proteases, here is allowing us to use multivalent interactions to inhibit the virus entry, and here to deliver a targeted payload to cells. And then this is one of the really kind of exciting things about being at MIT and being at the KI and working with colleagues at these interfaces. I'm going to thank you for your attention and I'm happy to take questions.
Wonderful. Thank you so much, Dr. Bhatia. It's really exciting to see like all the collaborations within the Institute that have contributed to all three of those projects within your lab. Maybe we can start with sort of an initial question about the diagnostic prognostic sensors. Have you tried adapting these for infectious diseases in the past? It seems you've been able to adapt a number of research threads in your lab. So just wondering if you could comment on how they've all come together in this way.
Yeah, that's a great question. So it's true, we have been thinking about the connections between sort of the microenvironments of cancer and the microenvironments of infected tissue for quite some time. And actually the use case that we had been working on was one that came to us from the Gates Foundation. And the idea there was if you have a patient that's short of breath, so they seem to have pneumonia, do they have bacterial pneumonia for which you should prescribe antibiotics, or do they have viral pneumonia? And so this was something that we were already working on. It was a funded project in collaboration with the Reagan Institute and Stanford, Pravesh Kathri's group, where we're looking at bacterial versus viral pneumonia. And that was kind of how we got into already having models of viral pneumonia going for this work.
Wonderful. And maybe as a follow-up, could you just comment on the sensitivity and specificity of detection of the cleaved fragments?
Sure. Yeah, so this is, of course, you don't know the answer to that until you get the data, and we don't have the data yet. In general, one thing that has been a kind of a happy surprise for this protease detection methodology has been the advent of AI and machine learning tools for us. Because actually, I describe this process like you're making one sensor, but typically we make about a dozen sensors. And we administer a cocktail. And then the way we approach it is we do a training study, a test training study, and then make a classifier, and then a validation study. And so that training study allows you basically to use machine learning and AI to create an algorithm that can have very high sensitivity and specificity, because you actually have a dozen different signals coming out. And so while I don't know yet, it's been very successful for other approaches.
Well, that's fantastic. It's very exciting. A question on the third project. Maybe if you could just give us a sense of what the target cells are for the RNAi, and then if you might be able to compare and contrast the RNAi approach with steroid approaches that have been in the news recently for blocking transcription factors or host response in general.
Sure. Yeah. So I think, I mean, I would say at the outset that all approaches are at this point welcome for blocking inflammation, right? So I think we're all trying everything. This is, I will say, as Angela did, this is, you know, a brand new early stage idea. So, you know, we imagine that it would take quite a while to bring to the clinic. I do think it would be potentially very broad, because it could block macrophage inflammation in a variety of disorders. And so macrophages are the target cell in this case, and so the targeting peptide that we're using actually targets activated macrophages, and that's how it works.
Gotcha. One last question from the audience. What is the fate of the polymer or linker after release, if any, from the reporters? Do those accumulate, or are they harmlessly broken down? Or?
Sure, that's a great question. I think that's about the biomarker, synthetic biomarker work, the diagnostic work. Yeah, I think one thing that's actually really exciting about being a technologist in this space is that these are kind of modular building blocks. So you get to decide what that polymer is in the middle. And so we've done things like make the polymer core something that could be imaged, like put a PET label on it or put an MRI contrast agent on it. So you could use it for something else. Or if you just want it to go away and be biologically inert, you can make it out of an inert polymer like PEG, which is FDA approved. And you can do things in between, like we've made some that are cysteine release so that they provide consistent monitoring over weeks. And so that's kind of actually the fun of it, is that you get to, it's a completely synthetic biomarker system. You get to choose what attributes you want it to have.
That's awesome, fascinating. I wish I had taken a stronger background in synthetic chemistry into myself now after hearing you and Dr.
You just
Kohler.
collaborate with us and then you'll have it too.
When we're fully ramped up, look forward to it.
That's right.
So with that, we'll move on to our final speaker of the day, who is Professor Bob Langer. So Dr. Langer is the David H. Koch Institute Professor at MIT, and a chemical engineer of great renown. As many of you probably know, he's founded a multitude of biopharmaceutical companies, and today his presentation will focus on one of his real signature areas of expertise, drug and vaccine delivery. So Dr. Langer, whenever you're ready.
Okay, so why don't we just go right to the, I'll probably talk fast, so we'll go to the first slide. And I was gonna just break what I'm doing into two areas. One, if we could go to the next slide. First, I thought I'd just give you a very, very fast overview of all the things we're doing that have relation to COVID, and then I'll go into a little bit of detail on two of them. So first, we're working, and some of these are with companies that I've helped start, and some are in our lab. So first, there's the area of vaccines, which uses a lot of nanotechnology, some of which actually got started in our lab and at the Koch Institute, and I'll mention some of that. But the major one is a company I helped start called Moderna, and they're in phase 2 trials right now for the COVID vaccine, and they will be in phase 3 trials very shortly this coming month. We've also been working on what I'll call self-boosting vaccines, and that's been with the Gates Foundation, so that you could only give yourself injections once, and I'll mention that. These two areas I'll go into a little bit more. But in addition, I've been advising a company, SIO2, to make new vials, which in fact, they've gotten $150 billion BARDA grant to make vials for Moderna and other companies. I've been working with my former student, David Edwards, on new nasal sprays that could hopefully combat COVID. We've worked with on several projects with masks. Our lab helped New Balance, which is a sneaker company, in the design of masks, and they've already made over 2 million masks that have been distributed to healthcare workers. We've also worked on split ventilators. In fact, several of our postdocs and students published, and we published in Science Translational Medicine, a way to make a very safe split ventilator. And then another of our postdocs, Syed, worked on actually a very cheap ventilator, a $200 ventilator. Also, we've started another company, 7 Sense, that is developing essentially painless way using microneedles for blood draws. And it's already been approved by the FDA for several things, and hopefully for COVID soon. And finally, we have a project with Lihu Sai at the Pick Hour, where we're making organs on a chip, brain organs, to study the neurologic impact of COVID. So with that's kind of an overview. There's also a few other things too, but let me just start at the beginning and go to the next slide. And interestingly, we're in a cancer institute, and the work we did that led to really much of nanotechnology in medicine today actually started with cancer research. I was a postdoc in Judah Folkman's lab trying to actually isolate the first substances that could stop blood vessel growth. And we needed a bioassay to study that. And that meant we needed to have little particles that could release molecules of any size for different time periods. And we created, this goes back to 1974, we created these little micro and nanoparticles. You see a picture here, and one cut in half. And we published, actually, in the next slide, a paper in Nature showing for the first time you could release molecules of any size in bioactive form for any length of time. Also in that same paper, we published the first time where we showed you could actually use microparticles and nanoparticles to deliver nucleic acids as well. Next slide. But I should point out that when I did this work, it met with enormous skepticism, both the angiogenesis work and the micro and nanoparticle work. I got my first 9 grants rejected. I'm a chemical engineer, and I looked for faculty positions. No chemical engineering department would hire me. And actually, finally I got a job in the nutrition department at MIT. And the only reason I think I got that was because the guy who was head of it, Nevin Scrimshaw, he was kind of like a benevolent dictator kind of professor, and he liked me, so he hired me, but he didn't ask anybody else in the faculty what they thought. And that would have still probably been okay, but the year after I joined, he left. So a lot of the senior faculty decided to give me advice, and their advice is I should leave too. At any rate, this is actually a quote from my friend Michael Marletta, who's in the National Academy of Sciences and was a professor at MIT at that time. And he was just recounting in 1980 when I proposed some of these ideas, and we went to a faculty dinner. This is a direct quote from a speech he gave at a Chinese restaurant with me and some senior MIT professors. He said, a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. And Mike said he thought he was in a Fellini movie. But we kept at it. And, and I got involved in patenting and starting companies, and I had tremendously great students at MIT. And today there are many, many products that are already approved and used by literally hundreds of millions of people based on these ideas. The next slide just shows a picture of one of those, of some of them. And they're used for cancer, for opioid addiction, for different eye diseases, schizophrenia, to prevent bleeding, lots of other things. These are just some pictures. But we kept continuing the work, and one of the issues with nanoparticles was making them not be gobbled up right away, and so we made nanoparticles. If we go to the next slide, that had certain types of coatings. This was from a paper of Science in 1994, and you heard Sangeeta talk about polyethylene glycol. So this is what we used, and I thought I'd show you a video that was made on Nova explaining some of this. Here it is. Is the sound working? Is the sound working okay?
Yeah, I just need to reshare it.
Okay, good.
That gets encased in a plastic that releases the drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
So that's an area we've continued to do a lot of work in, and those approaches can be used with polymers or lipids and to deliver small molecules. And actually, the nanoparticles have been used already at Alnylam, different nanoparticles to deliver siRNA. And Moderna, which I mentioned, is on something like 12 clinical trials, 9 for vaccines used for messenger RNA. Again, different types I should point out that all these companies have taken it far further than what we did, but we helped get it started. And now people make all kinds of different, lipids, different polymers, and there's just lots of exciting things going on in the nanotechnology area. The other area that I want to mention on vaccines started because I had a visit. I sometimes make this joke. I had this visit from a very successful computer entrepreneur, and that's shown in the next slide.
He starts with a
not this, yeah, there. This is my office at the Koch Institute, and that's me, and that's the successful computer entrepreneur, which is Bill Gates. And next slide. And Bill and his team have been incredibly generous to us. And one of the things that they were interested in, again, was vaccines. There are 25 million infant deaths. 10 million of these receive a dose of one vaccine, but they remain at risk. And what they really wondered is, could we come up with a way to give what I'll call self-boosting vaccines? You give one injection, but then you get these pulses at time 0, at a month, at 2 months, really at any time you want. And that would have 2 possible advantages. One, if people have poor compliance, and sadly in the developing world and even in the US, they have very bad compliance. People don't go back for a second injection. And in addition, the other big advantage is you may get higher antibody titers, which we've seen that in some cases, because you literally give one injection and you could have almost an infinite number of boosts. So we actually wanted to try to get this pattern, but we had to make the nano or microparticles in a different way. And Anna Jakulenik and others in our lab came up with a way. We started looking at different type of printing approaches, but none of them worked. So we came up with a new way of doing it, and that's shown on the next slide, which we published in Science about 2 or 3 years ago. And it involves 3 steps. I'm going to show you a video on it, but where you have, and it actually is almost involving what I'll call pico technology. It's even smaller than nanotechnology, but you start out with a mold, and the mold could be whatever shape you want. We then fill it with this biodot picoliter system. That's why I call it even pico technology. And you fill the core with the vaccine, and then you seal it. And the beauty of this is that by changing the polymer composition or the lipid composition and its thickness, you can make these burst at any time. It's almost like nano eggshells. And the shell you could make burst at time 0, at a month, 2 months, a year, depending on how thick you make that eggshell and the polymer composition. So let me show you another video.
Is
this working?
Yes, it should be.
Okay.
The stamped assembly of polymer layers, or seal method, combines computer chip manufacturing technology with a novel layer-by-layer assembling method to create an array of core shell polymer particles. First, the polymer of choice, PLGA in this example, is melt-pressed using a prefabricated silicone mold. The mold with the particles is then transferred to another substrate, where it is peeled off, leaving behind an array of polymer-based particles. These are then filled with any drug or vaccine using an inkjet piezoelectric nozzle and then dried. To seal the filled particles, a cap mold is aligned, sealed with the base, and delaminated. The resulting array of core shell particles are then removed from the base and stored until use.
We could go to the next slide. So Ann and her team, Kevin McHugh and others, they've literally made thousands of these. And here's a graph part of it from that science paper, and then additional work where we've simply changed the compositions. But notice that you get 6 different pulses by doing this. So you could make it go longer or shorter, and you could actually control the times by, like I say, the polymer composition and so forth. And you can give them all with the single injection. And if you, this is in vivo. The next slide just shows that if you look at dyes coming out, whatever happens in vitro also happens in vivo. And then we've actually looked at, I'll just show one example. This is really the last slide, at how well they work. And you'd lose nothing on storage. And if you compare just injection 2 boluses, 2 injections, to the core shell particles, we actually do better on that. And there's different controls here. So basically, this is a way where you could give many boluses, but just in a single injection with a potential for higher antibody titers, but without question having better compliance. I should point out, going back to what's going on with COVID right now, people, some of the vaccines, people get 2 injections, others they get 3 injections. At any rate, so this I think would have potential for not only the developing world, but lots of other places as well. If I could go to the last slide. Again, we've been very fortunate that this is my Cape House with my lab, and they're really responsible for all this stuff. I mean, they've done a great job, and we've been very fortunate to have funding from the Gates Foundation and NIH, and like I say, and I've been very fortunate to work with a lot of great people at companies, many of whom are my students or former students. Thanks so much.
Wonderful, Dr. Langer. Thank you so much. It's great to see the breadth and overview of all the work from your lab, and the portfolio of companies you've started has really been key. I was wondering if maybe you could comment on sort of the key lock polymer technology, the kind of almost looked like in eggshell encapsulating molecules. On what do you think its potential will be to specifically bind either the COVID-19 virus or virally infected cells?
Well, I think you could do that. I think that's certainly possible. And Sangeeta and others have talked about things that you could add to do that. But it's also complicated because you have cells like macrophages that grab onto things. And that's why we put the polyethylene glycol there. But the beauty of it for vaccines, which is what I was focusing on, is you don't even need to do that. You can just make a lipid nanoparticle that could either have adjuvant type properties or transport properties. And so for vaccines, you don't need to do that. Basically what you want for a vaccine is a nanoparticle where you can put the vaccine in, it protects the vaccine from, the RNA in our case, from getting destroyed, and then carries it into the cell. And then the cell actually makes, makes the protein. That's actually the beauty of messenger RNA. The reason why Moderna has gotten so far along on the COVID vaccine is that, there's a central dogma, right? That DNA makes RNA makes protein. So for years and years, what people have done is they use attenuated viruses. They might take eggs and make proteins. But here, and you need these giant factories and everything else and a lot of time to make it. But here, what Moderna has done, and now some other companies are doing that too, is you could take the RNA, and the RNA, and we have a big plant at Moderna out in Norwood, and the RNA will make, you just give that in the nanoparticle to the patient, and then the patient is the factory. The patient makes the protein, and then you get the antibodies. So that's actually, to me, a very powerful way of doing this, and I think that's probably why it's moved so fast. And there, the nanoparticle needs to have the right stability and shelf life and some other properties, but you don't need the lock and key thing.
I see. And so the Moderna vaccine is using this kind of this injection technology and encapsulation technology?
Moderna vaccine is in nanoparticles, right? So basically you take the messenger RNA, you put it in nanoparticles, and then you inject the nanoparticles with the messenger RNA into the patient. That's what's happening in all those clinical trials and hopefully beyond.
Wonderful. And maybe we could wrap up with a question. You're sort of widely considered a management wizard just for handling this ever-expanding portfolio of lab projects and company projects. With all of this kind of starting at once in the midst of a pandemic, was wondering if maybe you could comment on what challenges this moment has posed for you.
Yeah, well, I mean, the biggest challenge is I've had to get better at Zoom and things like that. But, you know, I think I'm on Zoom or WebEx or, I mean, there's so many different things that different people want to use. And I'm very, what I say computer challenged. I had to get my wife to come in and help me even on this. But, you know, really, it's just, I just feel bad for people, you know, both that we're not up at full capacity, you know, and yet I think people, you know, really, you want to make sure that everybody's safe. I mean, so I think it's a challenge for, you know, MIT, it's a challenge for the country, a challenge for the world in terms of sort of walking that line between being as safe as possible, which to me is the #1 thing, and still trying to, you know, get your work done.
Wonderful. Thank you so much, Dr. Langer. So in the interest of time, I think we'll kind of call it there. And thank you so much to all 4 of our presenters, really, for just highlighting the breadth of work across the Koch Institute and on COVID-19. And, you know, we've really appreciated our chance to share our COVID-19 work with you. I know everyone in the building is eager to return to our primary mission of cancer research. And, you know, as was mentioned, we've sort of just begun ramping that back up. And in addition, we're excited to, you know, make more progress on all of these new exciting COVID-19 projects and other ideas alike. So I just want to mention before we break, the Koch Institute programs team is putting together their fall events schedule, including the rescheduled Solutions with Insight, Pushing Boundaries and Breaking Barriers in Brain Cancer. So on November 9th at 4 o'clock, Dr. Yaffe will chat with Paula Hammond and Fred Lam about their shared work using nanoparticles to deliver therapies across the blood-brain barrier to treat glioblastoma. In addition, at some point this fall, there will be the Koch Institute Awards Gallery, where we celebrate the new images that have gone up in the lobby that we can now hopefully start to get back in and see again. The date for that is still in the works. So again, apologies to everyone whose questions we didn't get to. You know, we tried to pick what we could from the diversity of questions. Thank you so much for audience participation. And as always, you can subscribe to our mailing list as well as see the recording of the talk by visiting our website, ki.mit.edu. So until then, thank you all so much for joining us for this event. Please stay safe and in good health and in cancer research solidarity. Adieu.